Hello, student. This is Dr. Faizan Mirza. I'll be covering the questions related to mitotic cell cycle in this video. Okay, so students, this question came in Feb March 22, variant 22. The question asked us this cell is shown at the main stage of a mitosis, the mitotic cell cycle. What stage is this? Since you can see there is no nuclear envelope, the spindle fibers can be seen, and the spindle is holding the chromosome at the centromere, and the chromosomes are all aligned in the center of the cell. So this must be metaphase. How many chromosomes are these? These are a total of 4 chromosomes. Whenever you are counting the number of chromosomes, just count the number of centromeres, because all the number of centromeres that are there, that actually mean the number of chromosomes as well. So if there are four circles here, that means there are 4 chromosomes. So don't get confused with the number of chromatids and the number of chromosomes, because they change according to the cell cycle stage that the cell is in. So these are 4 chromosomes aligned in the center. Now, there they ask you, figure 2.2 shows the cell in figure 2.1 at the start of the cytokinesis. Cytokinesis is not the part of mitotic cell cycle. It's the part of cell cycle where the anaphase and telophase actually ends, and the cell starts to split. The organelles divide into two equally, and the cytoplasm starts to split as well. And the cell membrane, it starts to form a furrow, and it invaginates until the cell membrane join together and two separate cells are formed. Anyhow, cytokinesis, I was just recalling this. Complete figure 2.2 to show the daughter cell chromosomes in each of the nucleus. So you need to show the daughter chromosome. What will we do? We'll show 4 chromosomes. And again, 4 chromosome means just make sure that 4 centromeres are drawn, and around each centromere you will draw the chromatid which is there. So what I have done, I have not shaded any, because you can see that this chromosome is shaded, this chromosome is shaded, whereas these two are unshaded. So if there is a shaded molecule, shaded chromosome and a non-shaded one, since our board says that we should not shade our diagram, so I've avoided shading altogether, because shading is not required. What I've done, I've just drawn a longer chromatid and a shorter chromatid, because I've drawn two which will be long, and I've drawn two which will be short. And since each at the end of mitosis, both the cells end up receiving the same number of chromosomes that were there initially in the parent cell. So if it was having 4 chromosomes, so I need to make sure that both of them are having 4 chromosomes each. But the shape of the chromosome cannot be of this sort. It will be in the shape of single chromatids, because we know that in mitosis, by the end of mitosis, the sister chromatid, they separate, and each chromosome end up having a single chromatid each. That's what I'll be doing here. State the role of telomeres during DNA replication. It prevents the loss of genes from linear DNA. We know, and linear DNA, two linear DNAs can combine end to end, and telomeres prevent that as well. So furthermore, multiple myeloma is a type of cancer in the bone marrow where some of the stem cells start to produce abnormal cells. One treatment is to collect the stem cells from the bone marrow of the person with multiple myeloma. Healthy stem cells are isolated and grown in labs. So you take the healthy stem cells, you grow them in lab, and when you grow them in lab, they will divide by mitosis to produce a large number of normal cells. Because all these normal cells, once grown in lab, can then be given back to the patient of multiple myeloma, so he or she will be cured. Radiation is then used to destroy all stem cells which are cancerous. Finally, large number of healthy stem cells grown in lab are returned to the bone marrow. So these are 3 steps in treatment. So suggest the role of stem cell in treatment of multiple myeloma. So what's happening here? Stem cells, they have the ability to divide by mitosis. They supply cells that can differentiate to form specialized cells, as well as replace the use of stem cells. These grow in lab and can be put back into the patient, where they can divide by mitosis and provide a patient with the required number of cells. They can produce any type of blood cells too. So the person with multiple myeloma will be helped in this way. Let's move to another question. Okay, so this question, okay, so here we have this question, and this question actually came in October, November 2017. And the variant that I'm taking this question from, it's from the first variant, variant 21. So the question here, it shows us how an a photo micrograph of plant cells showing different stages of cell division. So let's just study this diagram first so we can answer the question. Okay, so you can see these are cells here, and these cells can be seen. So what stages do you see here? They are various stages of cell cycle shown to you. What stage are they asking? They are asking on the figure, draw a circle around a cell which is an anaphase. So anaphase cell means that the cell must have chromosome which have been pulled apart. So this is how the cell will be in anaphase. So you will just point out the cell which is having anaphase. So there's the advantage of using light microscope rather than electron microscope to study cell division. We know for electron microscope, the image, although is way more detailed, but it's always a dead specimen. Cell division is a living process, and electron microscope cannot be used to follow the living proceedings of a cell in real time. It can only be used. It can only be used to view details, and only dead specimen can be viewed. So that's the limitation there. So that's why we're using light microscope. Mitosis is important in producing more stem cell for plant growth. Describe three other ways in which mitosis is important in plants. So mitosis is used. You have to give three points why mitosis is important. So mitosis is used to produce genetically identical cells, and when they are genetically identical cells, they will be clones of each other, and these cells they keep on performing the same function. So when they keep on performing the same function, these cells they will perform the same functions as the cell they were about to replace. So the cell that was there and the cell that is about to come, they both will have the same function. So tissue will keep on having the same function. Mitosis also allow tissue repair to occur, and in this way, mitosis allow asexual production to occur as well. So yeah, all these points can be stated. Okay, so this question is from May June 2018, video 23. This is the micrograph of a root tip meristem, and some stages are shown to you of cell cycle. Cell D and E are in the same stage. Cell D and cell E. You can see cell D is in the same stage as cell E. So they're saying that they are in the same stage. State the difference between the nucleus of cell D and cell E. So what you can see here, you can see that cell D and cell E, they basically are having two nucleus here. So yeah, actually, it's not the nucleus, is a nucleolus. So I've just to make sure that you don't really fall for this trick, because at times the two term nucleus and nucleolus they appear to be similar where actually they are not. So here make sure that you know what a nucleus and what's a nucleoli. Even at times these terms, since they sound similar, so at times you get carried away when writing, and that's actually what happened to me as well. So cell D can be seen to have it's having two nucleoli. So these are the two nucleoli, and this is one nucleus here. And if you go to cell E, this cell E is having one nucleus, and there's one nucleolus, and this is the whole cell. And cell D is one cell, and this is the whole nucleus, and there are two nuclei within the cell. So name the stage of mitosis occurring at stage A, B, and C. So if you compare A, B, and C, A, B, and C, you can see in A and in C. If you go to A and if you go to C, and if you go to B, so what stages do you see here? What stages do you see at B? So in A and in B, you can see that the nuclear envelope is very well intact. So in A, yes, it's intact, but in B, the chromosomes have appeared. So, and the chromosomes have been pulled apart. This is how the chromosome get pulled apart. So in B, the chromosomes are still together. So they both are prophase. So A can be early prophase and B can be late prophase. Whereas C, in C, you can see the chromosomes have been pulled apart. When the sister chromatids have been pulled apart, so that need to be anaphase there. Cell FNGR, newly formed cells, cytokinesis occurred in the formation of a cell. Describe the events that have occurred in the stage of mitosis immediately before cytokinesis. So immediately before cytokinesis, we know what's happening. So we need to highlight what's happening just before cytokinesis, and that's what will be happening there. The chromosome would have moved to the poles of the cell. The nuclear envelope will reform around the cell, separated with the sister chromatid. The nucleolus will reappear. Spindle fiber will disassemble, and the chromosomes will decondense. That's what you'll be mentioning on the for this question. That's it from my side. I hope that is helpful for you. Thank you so much.